As Yona said, I'm Aaron Wenger. I'm Associate Director of Product Marketing at PacBio, and I'll be talking about the applications and the new updates on HiFi reads for characterizing human genomes. One of the really exciting advances in this last year was the application of HiFi reads to get really excellent contiguous and accurate human genomes. There was a there were two different HiFi genome assembly algorithms that were published. First HiCanoe in August of 2020, and then HiFi ASM in February of this year. And both of these algorithms are able to take advantage of the length and accuracy of HiFi reads to very quickly, for HiFi ASM in about 500 CPU hours, produce genomes that rival the human reference genome in quality and contiguity. And you see at the bottom here 4 different human genomes. So the GRCH38 being the international human reference genome that has a contiguity of around 58 megabases and extremely good accuracy, a complete size of 3 gigabases, and containing, by definition here, 100% of genes. But below that are 3 different HiFi ASM assemblies of different humans. So CHM13 being an effectively haploid human, HG00733 being a diploid female, and HG002 a diploid male. And in all 3 of these genomes, the HiFi ASM assemblies actually are more contiguous than the reference genome because unlike the reference, they're able to span some of the centromeres. They're extremely accurate with less than 1 error per 100,000 bases. They're complete human genomes, both in terms of size and in the number of genes that can be complete or that are included. So here it's now relatively straightforward with these algorithms to produce genomes that are reference grade. And also this year, the telomere to telomere consortium took this to its conclusion for that CHM13 reference, being able to start with HiFi reads, produce an assembly graph with HiCanoe, and fully resolve all 23 chromosomes from end to end, including the most difficult repeats in the centromeres. The consortium estimated that its final reference had an error rate of less than one base wrong for every 10 million bases in the assembly, showing that with HiFi reads, the length and contiguity, or the length and accuracy, allow you to get effectively perfect human genomes. And because it is a consistent result, you can also think about scaling this process, which is exactly what the Human Pangenome Reference Consortium is doing, moving from one complete human genome to updating the human reference based on 350 different human genomes. This last year released data from their first year of data production, which included 40 different human genomes, all diploid human genomes of diverse ancestries. And these genomes are all assembled with, or sequenced with HiFi reads and then assembled with HiFi ASM. But it's not only genome assembly for which HiFi reads are useful. It's been demonstrated now over by many different groups that having long reads from PacBio can improve the detection of variants in genomes. First with structural variants, where having longer reads improves the detection of bigger variants. This last year, the Human Genome Structural Variation Consortium sequenced 32 human samples from the 1,000 Genomes Project, both with PacBio reads and with other technologies, and demonstrated that with a typical individual with PacBio reads, they'll detect 2.5 times as many structural variants as you would with Illumina whole genome sequencing. And here, a structural variant is defined as a variant that involves 50 or more bases, primarily large insertions and deletions of sequence. But importantly, it's not only structural variants where long reads add the ability to more comprehensively characterize the genome. The Genome in a Bottle this last year updated its benchmarks using HiFi reads to cover more difficult regions of the genome that are not mappable with short reads. And for the reference sample HG002, that expansion of the benchmark added more than 300,000 single nucleotide variants and 50,000 indels, including many in exonic regions. The Genome in a Bottle, interestingly, also took the advantage of this ability to expand their benchmarks to launch what they called the Precision FDA Truth Challenge V2 to evaluate different technologies and different variant colors for how effective they are at characterizing human genomes. So as I mentioned, that the first benchmark that was updated was for HG002, but there are actually 7 Genome in a Bottle samples, and the benchmarks were updated for all of those. But rather than publicly releasing the improved benchmarks, Genome in a Bottle chose instead to hold back the benchmarks for the benchmark being the truth set for HG003 and 4, and simply release the sequencing data that underlied those benchmarks. Groups are then invited to take that sequencing data and analyze it with a software of their choosing, and to submit call sets, which could then be scored against the not yet released answer. And in this challenge, the single best one technology call set was with PacBio HiFi reads in the deep variant caller. Across the entire human genome, there were 13,067 total errors, which was 1.7 times fewer than the best Illumina call set, which was using the Dragon caller, and 6 times fewer than what would come out of a standard GATK4 best practices. Most of the improvement in the HiFi reads compared to the Illumina reads is in the ability to detect single nucleotide variants, driven by the ability of the longer reads to map to more difficult regions of the genome. So the HiFi reads bring the ability to map with their length and then to call variants accurately with their accuracy. And you can see the importance of that accuracy piece when you compare it to the bottom bar here, which is the Oxford Nanopore long read call set. So the best Oxford Nanopore call set, which also used deep variant, had 30 times as many errors as the PacBio HiFi call set, driven by primarily, so with more errors for all variant types, but particularly small insertion and deletion variants. Since the Precision FDA Truth Challenge, the variant callers have improved for many of these technologies. With the current state of the art today, deep variant now has 9% fewer errors than in the, or deep variant for HiFi reads has 9% fewer errors than in the Truth Challenge, so around 12,000 errors across the genome. And the Oxford Nanopore deep variant call set has also improved, but still has 21.1 times more errors than the HiFi deep variant call set, with huge numbers of insertion and deletion errors in the current state of the art. When you think about what HiFi reads add to variant detection in human genomes, it's, as we discussed, structural variants, especially those in the 50 to 1,000 base pair range, the ability to call mobile elements insertions, to detect copy neutral variants like inversions and translocations, to characterize repeat expansions, and then to characterize these single nucleotide variants and indels in difficult to map regions. And this general set of characteristics has inspired groups like the Genomic Answers for Kids program at Children's Mercy Kansas City, with whom Jonas mentioned we've been collaborating over the last year. This is a group led by Tomi Pastanen, and they chose to take samples from their cohort that were pediatric cases with rare disease that had not been explained by short read sequencing, and to see whether the improved ability of HiFi reads to detect variants could provide explanations for some of these cases. Among these 80 cases, there was a wide range of phenotypes, starting or with around a quarter of the cases having global developmental delay, but just as in any rare disease cohort, being a wide variety of different phenotypes. They sequenced all 80 of these cases and called variants, just as had been done for the genome in a bottle samples. But here, because these are real human samples, they were derived, or all the sequencing was from blood-derived DNA. Across this cohort, you see here on the left the number of variants that were called, in blue single nucleotide variants and in orange indels. You see it's very consistent results for all the Americans of European descent. And then you see additional variants called in Americans of mixed ancestry, primarily mixed African and European descent. But across this set, a median of around 4 million single nucleotide variants and 930,000 indels. These variants had the typical QC metrics you'd expect for good variant calls. And as these were samples from individual cases, not benchmarking samples, there's no true set against which to measure precision and recall, but you can measure concordance to other technologies. And for all 80 of these samples, the Children's Mercy Group also had short read whole genome sequencing and then Infinium global screening microarrays. So they were able to compare the precision and recall of the HiFi and short read whole genome sequencing single nucleotide variant calls against the microarray. And you can see the green dots here being the HiFi calls. They show higher sensitivity and higher specificity, so fewer false negatives and fewer false positives across this large set of samples of blood-derived DNAs, which is showing that the improved performance you see in genome in a bottle samples is generalizing to other human samples. Of course, single nucleotide variants are only part of the picture, and the HiFi reads provided also much better resolution of structural variants, nearly doubling the amount of structural variants that have been seen by this group with their short read whole genome sequencing pipeline, particularly adding an increase in insertion structural variants and deletion structural variants, and notably also decreasing the number of translocation structural variants, with most of the translocations called in the short read set being false positives. The Children's Mercy Group then interpreted these variants and reported a number of cases in which the HiFi reads were able to better explain the cases. This is one example of a 4-year-old female with hepatosplenomegaly, where the HiFi sequencing was able to identify 2 variants, one being a 4 base pair frame shifting indel, and the other being a single nucleotide variant. And critically, because these variants lied in a gene that causes an autosomal recessive disorder, having the HiFi reads was able to phase these variants and show that they were compound heterozygous, with one lying on one allele and one lying on another allele. So though these variants were 10 kb apart, with HiFi reads that are 15 to 20,000 bases in length, you're able to connect those variants and see that they in fact lie on different alleles. Another key advantage of the HiFi reads is the ability to resolve repeat expansions. And in one multi-generational family with dystonia, seizures, and ataxia, they identified a 1050 base pair expansion of an AT repeat that was intronic to STAR-D7 and segregated in this family with the phenotype. There have been many other groups as well that have been applying HiFi reads to look at human genomes in rare disease, with some great papers this year from a group at Nationwide Children's Hospital at Ohio State University, looking at individual with retinoblastoma, a group out of Hudson Alpha Institute led by Greg Cooper, looking at long read sequencing with HiFi reads of individuals with neurodevelopmental disorders, and then a group led by Namichi Matsumoto at Yokohama City University in Japan, applying HiFi reads to a cohort to identify a 12 kb copy neutral inversion in a syndromic intellectual disability case. And one thing we've noted is HiFi reads, they improve the ability to characterize genomes in many ways, both improving single nucleotide variants and structural variants, but also providing phasing and repeat expansions. And it's really, there are individual examples of each of these different capabilities helping explain rare disease cases. So it really is the more complete genome in many different dimensions that's improving the ability to explain rare disease cases. As we look to the future in human genomics, we really view HiFi reads producing really complete and accurate de novo assemblies and accurate variant calls, and serving as a platform for not only rare disease, which is mostly what we discussed today, but also a broader understanding of the human genome and being used to help predict someone's cancer risk or cardiac risk, or to understand their progression in neurodegenerative disorders. And we expect that this ultimately this one genome we carry with through life can be analyzed in many different ways to provide many different insights. And with that, I will turn it over to my colleague Michelle, who'll talk about the genome in plants and animals.